Hey everyone, today's video is going to be about mitosis and meiosis, and we're going to go in depth, and we're going to get into the nitty-gritty about prophase, metaphase, anaphase, telophase. So we're going to do that. First thing we're going to do though is we're going to introduce these terms of mitosis and meiosis. For mitosis, I want you guys to know that the main goal of mitosis is to make identical daughter cells to the cell that you started with, and that cell that you started with is the G1 cell in interphase before the S phase, so before DNA synthesis. The second thing we're going to talk about is how meiosis it makes genetically diverse daughter cells. So notice that one key distinction there between identical daughter cells for mitosis and genetically diverse daughter cells. These two main goals are kind of going to drive everything that happens that's different between these two processes. Lastly, just a quick little refresher on the cell cycle. So here we have the cell cycle, and I know it can look a little bit complicated because there's a lot of stuff involved, but just go around this way. It's like it goes in a circle. It's a cycle, right? So let's just talk about this right here. G1 phase is where a cell starts right after it's been born, and this is a phase of growth where organelles are copied. For our purposes, nothing really relevant is happening. There's this G1 checkpoint where DNA and organelles are getting checked to make sure everything's okay. That's not the purpose of this video. Then there's S phase. This is actually very important, so know about S phase. What happens here is that the chromatid number doubles, but the number of chromosomes does not change. So remember, during S phase, chromatid number doubles, but chromosome number stays the same. And this is true whether you're talking about mitosis or meiosis. And then you keep going along the cell cycle. G2 phase is more growth. There's a checkpoint here, and here you enter M phase. M phase is very important for our purposes as well. This is where mitosis happens, and meiosis is technically not considered part of M phase. It's kind of tricky. It's sort of an offshoot of M phase, and we'll talk about the differences and where they come up. And then notice that there's this other checkpoint called the M checkpoint there. So there's a total of 1, 2, 3 checkpoints. And lastly, you have cytokinesis. That's when cells actually like divide and pull away from each other. And then you also have G0 phase, which is a cell that's kind of resting. It's not doing much. So, okay, let's first kind of have an overview of the two phases, and then we'll get into the little details. So first of all, this is the cell that you start with. I'm going to flip this over. And this is a diploid cell. This is in G1, so this is before S phase, during interphase, right? So notice that there's a big A and a little a. There's 2 chromosomes here. Each chromosome contains 1 chromatid, so there's 2 total chromatids in this cell, okay? And then notice that this is 1 pair of homologous chromosomes. Different organisms might have different numbers of homologous chromosome pairs, but for simplicity's sake, we're just going to draw 1 pair of homologous chromosomes. So S phase happens, and it's the same whether you go into meiosis or mitosis, because these two processes are identical so far. What you've done is that you've copied each part of DNA from here identically. So now from one chromatid per chromosome, now you have 2 chromatids per chromosome. That's the same over here, and that happens for both chromosomes. So now you still have 2 chromosomes. The number of chromosomes has not changed. The ploidy has not changed, but the number of chromatids has doubled, and that's what happens during S phase. Number of chromatid doubles. Very important. Okay. Now let's look at the first phase of division and how they're different. So remember how we first said that, that's the wrong paper. So remember how we first said that the goal of mitosis is to make identical daughter cells, whereas the goal of meiosis is to make genetically diverse daughter cells. So here's where we see a deviation in these processes, and it's because of those reasons. So for mitosis, we want to make identical daughter cells, and look what we've done. We've separated this pair of homologous chromosomes that has 2 chromatids per chromosome, and we've taken 1 chromatid from each chromosome. So now we're back to, again, diploid cell, but it has not replicated. So you have 1 copy of each chromosome. So 1 chromatid per chromosome in each of these daughter cells. And then notice these are exactly the same as up here. So we've succeeded, and we've made genetically identical daughter cells. Okay, now let's look at meiosis. So there's 2 phases of cell division in meiosis, and the first one is really where things start to deviate from mitosis. So instead of separating the sister chromatids from each other and then taking one of each sister chromatid into the daughter cell, what you do during the first phase of meiosis is that you actually separate each member of the pair of homologous chromosomes. So here you see that one homologous chromosome went over here, the other homologous chromosome went over here. Now notice that these are now haploid cells. Why? Because there's no possibility for this to be like big A, little a, because it's the whole chromosomes here, the whole chromosomes there. So this is a haploid cell, but you notice that we still have replicated chromosomes, meaning that there's 2 chromatids per chromosome. Now, the second phase of meiosis, what it does is it splits up these chromosomes into their individual sister chromatids. So here we see that happening, and we've generated 4 daughter cells that are very diverse. So big A, and remember that due to the laws of independent assortment, when you add more chromosomes to this, you get many more combinations. So the point is that we've generated genetically diverse daughter cells from this one, as opposed to in mitosis, we've had the same identical cell. Okay, now let's get into the actual details. And I've made this chart right here. And you guys know prophase, metaphase, anaphase, telophase. Though the key difference between in meiosis and mitosis seems to be at the metaphase position. So first we'll talk about prophase. What I've drawn here is you can see that you have red and green chromosomes. So that's a paternal and maternal chromosome. These are a homologous pair of chromosomes, but you'll notice that it's unwound. That's because it's during interphase, when DNA is active and the cell is actually using the DNA to make proteins, DNA needs to be unwound. So during prophase, you're turning from this unwound state when DNA is active into an inactive condensed state, which is what you need when cells divide. So during prophase, all you're doing is that you're condensing the DNA into the form that looks like chromosomes as we know them. One key thing to remember is that we've passed S phase at this point, right? We're in M phase. That means that the DNA has already been replicated, which shouldn't surprise you when you get to metaphase. And during metaphase, you have 2 sister chromatids per chromosome. So you have 4 total chromatids here at the metaphase plate. So what's happening here is generally, by the way, you see this drawn as side to side, but actually think of these as one on top of the other, like the red and green are just kind of like that, as opposed to like that. But I've drawn it this way for simplicity's sake. And right now, the key of that positioning is that crossing over is happening here, and this is known as the synaptomeal complex. Okay, so that's metaphase. And then during metaphase, from metaphase to anaphase, this is where the key differences between meiosis and mitosis take place. So remember that during mitosis, we wanted to generate identical daughter cells. So what happens is that you're actually seeing those sister chromatids that we talked about earlier separating here during anaphase. Anaphase is a phase when, by the way, this over here, this is the mitotic spindle, these purple dots. And what they're doing is that this is a huge complex of proteins that are literally pulling these apart. Depending on either if you're going to mitosis or meiosis, they're pulling apart different things. So here we have anaphase. Anaphase, you're seeing the sister chromatids getting pulled apart, and then telophase, the cells actually divide. So you're seeing the genetic material during anaphase split up, and then the cellular material like the cytoplasm and all the extra stuff just lying around, that gets split up during telophase. And this was mitosis, right? And look, we've generated those identical daughter cells that we said we would. So let's talk about if at metaphase we went into meiosis. So like we talked about earlier, we see that it's each member of the homologous chromosome is getting split up. And then there's this phase, telophase, that is decondensed DNA for a little bit, and these cells might be active at this haploid stage for a while. They might not be, that depends on the species. But here you've seen the splitting up of the chromosomes. So paternal and maternal chromosome in a different cell. And lastly, if I can find the page, there's the page. So you have prophase, metaphase, anaphase, and telophase. These are very much the same as mitosis. So this was part 1, right? So this was mitosis 1 with anaphase and telophase, and you've ended up with these duplicated chromosomes in each cell. Now, essentially, it's just the same process where the same things happen. So DNA is going to condense again, and then the sister chromatids are going to pull these apart at the metaphase plate. And by these, I mean the sister chromatids, leading to, finally, these gametes at the bottom that are genetically diverse.